Okay, there's a question, how long will it take to develop resistance by the bacteria, antibiotic resistant? Well, antibiotic resistant is not a very fast process. Antibiotic resistant, it might take a long period of time to develop. Could be years, months and years. It need to develop the resistance. So we can't see on petri dish this kind of a resistant on antibiotic. Now, if I say, now we discuss the result, then I ask here, these two, antibiotic P. So one are coming like, if you bring the argument like antibiotic resistant, uh, you're going to say P is resistant on disk 1, and P is not resistant on dish 2. Rather than this argument, if I ask you dish 2, from the beginning, there are very less amount of bacteria around it, because we started from this like this. When you allow them 24 hours, it's completely filled with the microorganism like the blue color one. Now in the Q, you can again take now Q. Q, we didn't say something like that. We didn't see something like that. You should also consider Q, because Q, no difference. So what if I say Q, when they place after 24 hours, this 1, so we have quite a good number of bacteria, and they place the Q, this Q here, and it killed the bacteria around it. It killed the bacteria around it. And here, did not kill. And here, it prevent growth. Now give me one word, how you can say the antibiotic P. What is the correct name you can give for antibiotic P?
Bacteriostatic.
Yeah, that's a P is bacteriostatic and Q is bactericidal. So we prepare for this kind of tricky questions. You completely forgotten bactericidal and bacteriostatic. At least you can check Q, it killed all, and P killed these two. It killed here, and killed. So then the answer is antibiotic Q is bactericidal, antibiotic P is bacteriostatic. So that's why this one, it does not kill the bacteria, but it just keep the bacteria on P. It's not grow, but it doesn't increase the growth of the bacteria. Bactericidal and bacteriostatic. Okay. So we discuss immunity, infection, and this, and this, and this, and this. Okay, another thing I should highlight here, if photosynthesis asks, Celvin cycle, it is better we revise quickly about Celvin cycle. So here RUBP, and this is GP, and this is a GALP, and this is a carbon for intermediate product. Uh, GALP is important to produce glucose. Then glucose can be used to produce other type of sugars like sucrose, maltose like sugars, and also starch. So glucose play a big role to produce a whole set of carbohydrate. That's one part of the glucose, and the second part of the glucose can enter into the respiration. During respiration, the uh, uh, Krebs cycle and glycolysis, there are a lot of intermediate products formed. Then some of the intermediate products used together with nitrate from the soil to make amino acid. Amino acid make the proteins. And some of the intermediate products, just like acetyl CoA 2 carbon a compound, acetyl CoA from these link reactions, again respiration, intermediate product respiration, and with GP, and this one can produce fatty acid, and they form triglyceride, a lipid. And then phosphate from the soil, and then carbon 4 products from the Celvin cycle together can produce DNA and RNA. This is very important thing, and if you further ask different thing like how can photosynthesis happen in the leaf, can produce uh, starch in the roots. Photosynthesis happen in the leaves and it produces glucose. And how can starch produce in the roots to store? Then again, glucose produce sucrose. Sucrose is a transport sugar, and then sucrose can transport to the roots, and then root it can produce again starch inside the roots. So probably if you completely not aware of the you need two certain things like the transport in the phloem by sucrose is very important to bring sucrose to the root to make the starch like things. So just this one is a bit important, always keep in mind photosynthesis type of questions ask, these informations are valuable to write answers. I think I just revise what I want to say here, rather adding so many informations. If you have points to ask, maybe you can ask.
So, can you just revise the percentage increase and decrease? How to calculate?
This is Dev, right? I think I could hear the voice there. Devja, you? Yes.
Yeah, sir.
Okay. Because electronically the voice is not the same as. Okay. Yes. Okay, take this. Now percentage increase, percentage decrease, percentage change. If we ask any of these things, percentage increase, decrease, or percentage change, then for all, first thing we take difference. For example, this is the first percentage decrease, this is percentage increase, and anything between this change, this is change. So we take this difference, let's say D, divided by the starting value, initial value is this. Percentage decrease, again we take the difference, that is D, this one, and then initial value is this time, this, right, initial value 2. We take this as initial value 2. That means the difference is not changing. The initial value is changing. As a result, here we have a different number and here different number. If you have percentage change, theoretically, both could be okay, because we don't know increase or decrease. We can write initial value in many different ways. So change is usually okay for both. Not much worry about the initial value. You should be very careful about the initial value when you calculating the percentage change. Carefully select what is the first value. That's only one you should keep in mind. Right. Apart from that, any other questions? Okay. I have another question. I didn't want to take in a bigger way. I just okay. There's another question. Histogram and the bar chart. Okay, let's take one example, height of the people. When you take height of the people, we can find many different height of the people. But still, there is a number, ending number. For example, this is the short area, this is the tall area. So we can find people height in these two levels. But we know short people are less and tall people also less. So if we count in the population, how many people short, so we can come with frequencies like this. So then in the middle, I mean, I should have a same thickness of the bar, but just like this. Okay. So if we line up all the frequencies of the short and tall people in the population, and this each bar is attached. That means it's touch the other one, and this become now histogram like this. But when we are drawing a bar graph, we should keep a fixed distance between these two bars. It's important to keep some distance. This is two bars. We are not going to attach to all. So this is a histogram shows these frequencies or different numbers of some kind of one parameter, that is height. So one obvious feature of the histogram is the bars are touch with each other, and we see mostly a pattern like this, bell-shaped curve. Not always, but most of the time we can see this bell-shaped curve. But bar graphs, the height may be different to place, and they are not touched with each other. So this is important. Quickly identify whether this is a histogram. If it is histogram, then you have to look different way of writing or answering question. The bar graph is common. We know how to answer bar graph or draw bar graphs. Okay. So HIV is a human disease, and this human disease transmitted to human from monkeys. That means monkeys, they also have another similar kind of virus called simian immunodeficiency virus, SIV. You know, always we see this kind of a diseases if the animals there, this animal has virus inside them. Probably this virus is not pathogenic to animals, and some it is pathogenic to these animals. If there's a mutation happen, this mutated virus move into the human, then new diseases come into the world, like HIV. So HIV came from the monkeys, and monkeys had this simian immunodeficiency virus, and human has human immunodeficiency virus after mutations. To investigate about this kind of a thing, the scientists, they use kind of a test strip, like this kind of a strip, and this strip has a HIV antigen. They have HIV antigen. So different antigen are placed in this strip. So most of the diagnostic kit develop like this. One thing we can check, like COVID-19, we had this rapid rapid antibody detection kit or something like that, rapid antigen detection kit or like this, developed based on antibody antigen reactions. If we can get antibodies from the blood, then this antibody can bind to this antigen. Antibodies are specific. Now this antibody can bind to this antigen. After that, they make a second antibody to attach to this antibody. That means second antibody specific to the first antibody. Second antibody specific to the second antibody, and it is labeled with a color like this. Then it should have a color, okay, like this. Now this information is important because you can see there's a past paper questions. The plate is given, and the plate is called ELISA plate with 96 wells, and they had antibiotics and different bacteria like that, and they check many things. It is based on some of the tests based on antigen and antibody, because antibody and antigens are 100% specific. So if we attach antibodies to this strip, we take serum sample, plasma sample from patient, just add this to the strip. If antibody is specific, blue color one bind, then we add second antibody with the color label. So each step we need a washing step, because washing remove unbound antibodies. If it is bound, washing cannot remove it because very strong binding. It's a chemical bond, so it stay there. So this is the basic method to develop this kind of a test and it is very important for many questions. This concept not learned as a separate thing, but in the questions they say this is like this. Example like this: test strips were prepared which contain several different HIV antigen. When samples are applied, that means sample containing antibodies, to the test strip, a line will appear in the control region. If the sample contains antibodies to the HIV antigen present on the strip, additional line will also appear. Now, why I just show this one? We avoid uh, using too much time to understand this kind of a questions, because you need to read again and again to understand the question, which takes time. Now, if you know this one, okay, many diagnostic kits based on antigen antibody specificity and their binding. So here, then we can learn it. If antigen is there, then antibody binds, and we can add second antibody with the colored label one, and we can detect this color if it is bind. The binding are specific and strong. Each watching step remove unbound one. Then only the antigen antibody reaction happen, then we can see. That means specific. Here suggests why the investigators collect the fecal sample were prepared and then applied to the test strip. So monkeys, they collect fecal samples and they use these fecal samples uh, for this experiment. Suggests why the investigators collect fecal samples from chimpanzees rather than plasma sample. So what do you think? How do you think to answer this question?
It's easier to obtain.
Yes, we can say easy to obtain. Uh, it is very risk collecting a blood sample from monkeys, because if the monkeys are infected with diseases like SIV, and maybe who knows that this kind of viruses are sometimes harmful to human, like contaminations, and collecting the monkeys and then prepare them to collect the blood sample, and these things are very difficult to do and there's a risk. So easiest thing is collecting the fecal samples. Now, animal studies, we collect most of their sample. That means then are there antibodies in fecal sample? Now that is not a question for us, because if they say antibodies bind to the antigen on the strip, then there should be antibodies in the fecal sample. So these antibodies somehow coming out from the blood and then intestine, maybe they're adding to the transfer to the intestine. So then antibody should be there. So then we are not going to bother about too much thinking. You go for the best option. Okay, monkeys, maybe animals difficult to collect samples because it's a risk and time consuming. So always we take a blood sample which led from the trees and on the footprints, fecal samples, hairs like thing we collect. Okay, that's right. Now they collected 608 fecal sample from 224 individuals chimpanzees. Suggests a lab method that could be used to identify whether two fecal sample belong to the same individual or not. Now, this is something like this. When they collect the fecal sample is not labeled with the name of the individual, because fecal sample are same. But how they know that these are 224 individuals?
So gel electrophoresis.
Yeah, we learn again and again. We can identify individual difference, species difference, and similar species by gel electrophoresis. Because if they take these 6 or 8 samples and they perform gel electrophoresis, and samples are, the banding patterns are same, then they place as a one individual. And they have seen only 224 different banding patterns. Rest of them are matched with the certain things, like it's like a replica. So it's a replication of existing thing. So method is gel electrophoresis. I think based on the marks, you can write your answer. Think about still you need to write amplification of the DNA sample using PCR. Is it necessary or not? So you have to think about this. The second one, you write the protocol of gel electrophoresis based on the marks number. DNA sample are digested with restriction endonucleases loaded onto the gel, run the gel, and compare the banding patterns, similarities and differences. Similar bands belong to same individuals. These are the samples applied. Human plasma sample from human infected with HIV. Human plasma sample not infected with HIV. Fecal sample from chimpanzees infected with SIV. Fecal sample from chimpanzees who are not infected with SIV. So if they add all these samples, they see pattern like this. Human plasma sample, HIV positive, you can see the tested region is this region. Here, these are the antigens, all give positive result when it has antibodies for HIV. And person they don't have HIV, then no antibodies. We don't see any bats. And chimpanzees with SIV, they had at least 3 antigens which is shared with HIV. So then negative one, that means no SIV, then we don't see antibodies. Conclusion is test region HIV positive sample has 1, 2, 3, 4, 5, 6, 7 antigens positive, and SIV one shared only 3 antigens with HIV from the samples. Okay, one more question from this. Now, HIV infected person produce antibodies, meaning immune response there. That means cellular and both humoral immune response there. Why this response cannot cure HIV from this person? So you can understand your knowledge about the immunity. The end stage is antibody formation. Even antibody produced at the end. What is the reason these people can't live longer and they die because of HIV?
So because HIV virus attacks the T helper cells, which releases cytokines.
Yeah, it's a important thing is T helper cells. It is not that the antibodies should be there, but T helper cells should be there from the beginning to the end, because T helper cells drive all the process. If there is a war zone and at the end we see they can send 1 or 2 missiles, launch missiles. Missiles are powerful. But you know, all the important soldiers are no longer there. Only a few soldiers here. They send the missiles, but majority they are no longer there to drive the process. So T helper cells, CD4 cells need to the end of this process. So when T helper cells are no longer they are decreasing, then less B cell can activate. At the end, B cell also decreasing, and T killer cell also decreasing, because they are not increasing, their activation is not there, and they can't go further. So that's why even antibody produced T helper cell role is no longer there. Okay, I think and that's something other presentation like numbers. It's not that much great value for this question. I just want to have this question at the last. I'm all done. So still, do you have any questions? Otherwise, I wish to say good luck for you. And some of them, I think, already in better performance, because 1, 2, 3 are done. I mean, 4 is another number for them. But those who are having break can start again. Unit 4 may be a bit excited again after break. But don't be panic. Manage your time. Time management is very important. Do all the strategies to manage your time. Watch your time and check when you're writing how many questions finished and how long took it to finish, like that thing. You can check in the middle and maybe a little bit later as well to manage the time. And if you don't have any more questions, relax. You can't do much. Just keep in a very relaxed mood now and try to revise still very important definition like things. And I wish you all the best for tomorrow. You need 4 exam. I hope this class helped you to increase your psychological fitness, and you will do better, right? Okay, do you have any questions?
Sir, can you go through the climate model just once more?
Yeah, climate model, what I suggest, try to remember the facts. That means climate model is a collection of data and a graphical representation. That's the first point. Second point, climate model can be used to extrapolate to predict future climate conditions. Third point, it has a correlation, but always keep in mind correlation is not causation. You can say correlation, but you can use it is not a causation for certain cases where necessary. And climate models can be checked with the other available calibration method to make it validate. And the last thing, prediction can be changed, which is estimation, because we try to reduce global warming, so future it could change, or some of the activities can increase global warming. So that's all about the climate modeling. You need to remember.
Okay, sir.
All right, then I wish you all the best for tomorrow's paper, and we'll meet again Sunday for Unit 5 and 6 to finish it off. And have a good time and good luck for tomorrow. See you then.
Thank you,
Thank you,
sir.
sir.
Okay, bye.
Good
Take
night,
care.
sir.
Thank you, sir. Thank you, sir. Bye, sir.
Okay, bye.